Okay, southern blotting. Well, we've already talked about northern and western blotting, and as it turns out, southern blotting was the original. Southern blotting uses restriction enzymes, or restriction endonucleases, enzymes that cut double-stranded DNA. You're going to separate these fragments by gel electrophoresis and stain bands to make them visible. The blotting technique is going to consist of transferring DNA bands in a gel through an intracellulose paper or onto nylon paper. The hybridization, you're going to add a probe specific for a gene of interest that binds to a specific DNA sequence. So generally this is going to be radioactive, or you're going to use a fluorophore label. This is basically the exact same thing as what we talked about with northern blotting and western blotting in terms of the technique. You do a gel, a transfer, and then after that transfer you do a incubation with a probe, and then you detect. Between each one of those steps you're going to be doing a washing of those. You do not have to do any sort of blocking with a southern gel like you did with a western blot, but each one of these steps does require a washing. This technique is done in order to determine specific sequences of DNA within a complex mixture. So think about this as blood or tissue sample analysis. You can say whether or not even a single nucleotide differs between one sample and another because you've designed a probe that is very specific and binds only to one specific sequence of DNA. This technique is quantitative. It shows the amount of digested and undigested DNA molecules. It can be used in a quantitative fashion to analyze gene amplification. This technique was developed at Edinburgh University in the 1970s by Edward M. Southern, and it was named after him. This was the first of these blotting techniques, and this was kind of used as foundation for, well, analyzing DNA. So then it was like, okay, well, what can we do with RNA? Well, that's where the northern blot came in. What can we do with protein? Well, that's where the western blot came in. And then the fun thing is, if you were talking about probing for a protein and RNA at the same time, that's a northwestern blot. If you are probing for DNA and protein at the same time, that's a southwestern blot. This was published in 1975. Now, impact on the non-scientific community, the southern blot and RIFLIP analysis. This RFLP is restriction, fragment, length, polymorphism. That's what RFLP stands for. Analysis are used in DNA fingerprinting. So the crime scene analysis creates a pattern based on an individual's unique DNA. So you can this can identify a single person based on any sort of restriction fragment length based on polymorphism. So, one person has ATGC, and another person has ATTC. Well, these are different enough that whenever you cut them with a restriction endonuclease, maybe this one will cut, and this one will not be cut. And so you would see this result in a southern blot, or see the difference when you're probing for these two different pieces of DNA in a southern blot. This is also helpful in detecting genetic diseases and cancers. It was helpful in detecting sickle cell mutations as well as monoclonal leukemia, and detection of gene families in a genome, in an entire genome. This technique is used to detect one or more restriction fragments that contain a gene or DNA sequence of interest. This is used to study gene mutations, deletions, and duplications that could cause diseases.